All right, it is 2, so we will get started here. Just have a little to finish up from Friday. Then we'll go right into what we're talking about today. So just as a review, on Friday, we were talking about glycoproteins. And then we were talking about the peptidoglycan wall. And so we more or less ended up on this slide where we just looked at the basics of the peptidoglycan layer. And so I'll go through it really quick again. It's basically made out of 2 sugars, N-acetylglucosamine and acetylmuronic acid. That is going to be your dimer. These are connected to peptides. Usually that's a tetrapeptide. Doesn't have to be. When you research this, you actually see that it could be a pentapeptide, or there could be only 3 amino acids. It basically varies. Something interesting about these amino acids is that you can find D amino acids. And then these peptidoglycan layers, they will cross-link. And so the outer cell wall of bacteria is basically this sugar protein structure that's full of random holes. So this is the slide we ended up on, and that's just a little reminder about the topic. And so we're going to start with just some questions to make sure we have this in our head. And I think I should have polls for this. Yeah. So talking about the peptidoglycan layer, which is not a structural component of the peptidoglycan, and which feature is not essential for the peptidoglycan layer. I'll do about another 30 seconds here. All right. So what is not a structural component of peptidoglycan? And the majority of you got it right, sulfate. So if you look back on the previous slide and you look at the structure, there's not really sulfate groups in there. So sulfates aren't really part of the peptidoglycan. While a disaccharide unit was, we have acyl groups and we do have a tetrapeptide. Which feature is not essential for stability? Why do you put L amino acids? Which so the correct answer is D monosaccharides. Let me just go back to the previous image since the majority of us got that wrong. Although there is a D amino acid, we do have 3 other L amino acids. So the majority of the amino acids are L. But we don't have monosaccharides in here. We have a disaccharide, and they're not D either. We didn't really go onto that. Yeah, the correct answer for number 2 is D. There's no D monosaccharides in the peptidoglycan layer. Questions about that? Rather straightforward. Right. And so our last question to do with the peptidoglycan layer is what's the point of the pores? And I answered this on Friday at the end of the day Friday, so I'll just say it again. The pores are really letting molecules in and out, right? And so that's why this thing is just not like a solid wall, because this is on the very outside of the bacteria. So you have to let materials in and you have to let materials out. So that's why we have holes of just like varying sizes. B is more of a critical thinking question, one of these critical thinking questions. Why does peptidoglycan layer have D amino acids when every other amino acid basically on Earth is L? Why do we find D in this location? And I'll leave that question for you to try and answer. Differentiate as well. So got a large variety of answers there. My thought would be is that if everything is programmed to work on L amino acids, like all the enzymes that we have are supposed to see L amino acids. So if the very outside of you, the thing that would interact with foreign molecules, if you have D amino acids, that's one way to protect yourself, because enzymes aren't used to seeing this chirality, so they will go and try to eat it, and they will get stuck. They just won't be able to work. So you can think of D amino acid as one way to, as a safety mechanism to prevent the peptidoglycan layer from being destroyed. Of course, when that happens, the organisms that want to destroy peptidoglycan layers, their enzymes will evolve too to be able to actually interact with the D amino acid. So it's basically a war there where I change something. Well, I'm going to change that so I can attack that something. And we see this in bacteria all the time. Like we have an antibiotic, bacteria become immune to that antibiotic. So we have to create a new antibiotic that they become immune to, so on and so forth. So the short answer is to prevent the peptidoglycan layer from being destroyed or being attacked by enzymes. Questions about the peptidoglycan layer before we move on to lipids, our final major macro biomolecule in this class. Speaking of which, there's only 1, 2, 3, only about 5 weeks of class left. Really flies. The purple in that picture are crosslinks. That is cross-linking. And what that probably is the way that these things are cross-links is through an isopeptide bond, where it's lysine will form a peptide bond with, usually it's through another isoglutamyl, right? So that's why it's trying to show that here, where you have like different spheres being connected. Do they have a specific name? I think it's isopeptide bond, but they don't have like a name that's only for peptidoglycan. No, it's just it's a common feature that's seen in other molecules, so they get a common name. All right, so let me move on to my next PowerPoint then. Hey, we've caught up. So our last biomolecule of the semester are fatty acids, lipids to be more exact. So what is a fatty acid? So a fatty acid has a carboxylic acid and then a long chain of hydrocarbons, or just carbon and hydrogen. Most fatty acids you'll find in biological systems are 16 and 18 carbons long. Anything less than 14 is going to be uncommon. Anything greater than 20 is going to be uncommon. We do have those, though. They're just rarer than a 16 or 18 long fatty acid. No PowerPoint on Blackboard? Give me 1 second. That's an easy fix. Da, 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 da. Yep, sure isn't. Yeah. All right, should be there now. If you just want to refresh in the test 3 folder, it should be right there. All right, so yeah, fatty acids. So with our fatty acids, we do have common names: palmitic, oleic, linoleic, stearic. I'm not too worried about knowing those common names. I'm more worried if we can look at any fatty acid and give it a systematic name with numbering carbons. So the way that we count carbons, right, is that you start with the carbonyl carbon. You call that 1, and then you just count down, right? 1, 2, 3, 4, 5, 6, 7, all the way down. And what you do is that when naming your compound, you use this system where you say, how many carbons were in total? Well, for our first fatty acid, there was 18. You then put a colon and you tell me how many double bonds they were. So that's double bonds, number of double bonds. And this is the number of carbons. Right? So in our second molecule, oleic acid, we have 1 double bond. So that's why we have 18-1. And this delta 9 tells me where my double bond first starts. So our double bond is first on carbon 9. So this is an 18-1 delta 9 fatty acid. Our next example, linoleic, is 18-2 because we have 2 double bonds. So that's an 18-2 delta 9 12. And then lastly, we have 3 double bonds, 18-3 delta 9 12 and 5. Now, you can also, and this is weird, but you can also start counting carbons in the reverse direction. No, you only count carbon-carbon double bonds. So question, you don't count the double bonds in the carbonyl group? No, because that's C double bond O. We only care about carbon-carbon double bonds when I say double bonds. Right. So another way you can count fatty acids is starting from the last carbon and working your way up to the carbonyl carbon. If you do that, you have to call them omega. So if I'm starting on linoleic acid, that carbon is omega 1. The next is omega-2. The next is omega-3. Now, if you have a double bond at the omega-3 carbon, we call that an omega-3 fatty acid. Those of you who like reading about health and nutrition, you've probably heard of omega-3 fatty acids. Well, that's what it means. It just means you have a double bond on your omega-3 carbon. You also have omega-6 fatty acids. So here's omega-1, 2, 3, 4, 5, 6. So this is an omega-6 fatty acid. And I guess even though you don't really talk about them, technically this would be an omega-9 fatty acid. But usually people only talk about omega-3 and omega-6 when they talk about nutrition and health. Now, when talking about fatty acids, we have some terminology. We have saturated. If you're saturated, that means you have no double bond. So when you think of saturation, that means you're saturated in the maximum number of hydrogens that you can have, right? So saturated is no double bonds. If you're unsaturated, that means you have at least one double bond. Because you are not saturated in the amount of hydrogens you can have. So some questions here. What is the purpose of counting it backwards? So you can call it omega-3. What's the actual purpose? I do not know. I've never studied fat. So for some reason, I think it is. Okay, let me back up. I actually do know the reason. And the reason is that certain mechanisms, certain enzymes start from different areas of the fatty acid. Some enzymes start from the carbonyl head. Some enzymes start from the bottom, from the tail. And based on where the double bond is, your mechanisms change and what enzymes can work on your fatty acids change. And so it might be easier to say, you know, I have an omega-3. That means I have a double bond 3 from the tail. Instead of saying I have an 18-1 delta 15, which means the same thing, but it's way harder to think about. That would be my thought as to the reason why we have two different systems. Is that the true answer? I have no idea, but that makes sense to me. Is there a preference for either convention over the other? I think generally you're going to see fatty acids that's like 18-2, delta 9, stuff like that. But you will also see the omega version that will pop up. So I think the counting from the top is more common, but the omega system is not uncommon. Just because, like I said before, there are a lot of enzymes that start from the last carbon and work their way up. So it's just easier if we start counting from the back sometimes. Does the omega system count all the double bonds or just the first one? When you name a fatty acid, usually you just say like the first double bond. So this is an omega-3 fatty acid, right? So if we look at linoleic acid, you could say it's an omega-3, an omega-6, and omega-9, but usually you just say this is an omega-3 fatty acid. Because again, like I said, I think it's due to the enzymes that work from the tail backwards. So those enzymes, they go through different mechanisms and you use different enzymes based on where you encounter that first double bond, based on what chemistry you're doing. And if you're interested in that type of stuff, we cover that in Biochem too, about all the different enzymes that work at the different parts of the fatty acid. All right, so some more just common things about fatty acid. Your first double bond, usually at carbon 9. As you can see here in our 3 examples, they all first appear at carbon 9. If they're polyunsaturated, that is, if you have multiple double bonds, they're usually always going to be spread 3 carbons apart. So 9, 12, 15. And these double bonds are usually cis double bonds in that the CH2s are usually on the same side. So they're usually not trans, they're usually cis. And lastly, why unsaturated fats are better for you health-wise is because they can't pack closely together. So if you have saturated fatty acids, their tails can pack nice and close, kind of like I'm drawing, and you'll get strong non-covalent interactions. Think like Velcro. It'll be harder to separate those, and so these will form a solid more easily. Think beef fat or any animal fat, really. We have a lot of saturated fatty acids, so our fat is super hard and forms a solid because our tails are packing together. But with unsaturated fatty acids, I'm going to pick a different color for that. Unsaturated fatty acids, because of their double bond, they have a kink. So because of that kink, they can't pack. Actually, the kink is more pronounced than that. Let me do a better job. The kink is way more pronounced than that. It's like something like that, something like wild, where you just get like a tail going off at a 60 degree. And because you have that big kink, your fatty acids can't pack as well together as if they're saturated. So your unsaturated fatty acids, your olive oils, your other plant fats, they're more liquids at room temperature. That's why if you're cooking, it's usually better to use olive oil than it is to use like lard. Just because when it goes inside your body, that's going to make a big difference if those fatty acids can pack closer together or not. All right, so is there any questions about our general introduction to lipids? Okay, so let's move on. Question 1. Please tell me the names of these 4 lipids and tell me if they're saturated or unsaturated, and tell me if it's an omega-3. If you want to, while doing this, I'm going to give everyone a couple minutes. If you want to check your work, just send me a private message instead of broadcasting it to everybody, and I can say yes or no. Otherwise, I will give the answers in like 2 to 3 minutes, something like that. But yeah, this is now your time to practice what we just talked about. So I want the delta. So give me like the X dash Y delta form. And the omega-3 is either going to be yes or no. For the omega system, usually you don't use this notation with the omega system. So this is not omega. Like I said, the omega is usually only saying if it's an omega-3, omega-6 fatty acid. That's usually what you only use it for. But I want like 18-2, delta 912, stuff like that. So double bonds usually don't start at 8. So you might have miscounted a little bit. And I'll do A, just actually, yeah, I should have done A. So just to make sure we're on the same page here. So what I do is I usually just count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So we have 18 carbons. We have one double bond. That double bond starts at 9. It is unsaturated because we have the double bond. And it is not an omega-3 because there's no double bond at the omega-3 position. And I will just do omega, so we have that as well. This is omega 1, omega 2, omega 3. No double bond there. So there's A. Yeah, there is D hiding out in the corner. Okay, so let's do B since we're getting some answers. So B is a 25 delta 1, 2, 3, 4, 5. So this one starts at 5. So this is a little bit of an oddball, but 5, 8, 11, 14, 17, unsat. It is an omega-3 because we have a double bond right there at the omega-3. So yes, this is one of those healthy omega-3 fatty acids that you get from fish. Next one. So fatty acids don't always have to start from the left, right? This is carbon number 1. So make sure that you start from the carboxylic acid as your naming scheme. So C, I have as an 18, 3, delta 9, 12, 15. Unsat, it is an omega-3. And then for D, I have that's an 18,2, delta 9, 12, unsat, no omega-3. All right. Any questions? So yeah, D would be an omega-6. Any questions about our general naming scheme of our fatty acids? Yeah, so in reality, unsaturated fatty acids are like super bent. So that's what D is showing. That's what it would actually look like in reality, instead of like nice straight lines in A and B. Yeah, I mean, naming fatty acids aren't hard. You just have to get used to it. So for practice, what you can do is just like type in Google, fatty acids or molecular fatty acids, and you'll just get random pictures of like structures like this. You can just use that to name them if you wanted to. All right, so we're going to move on from this topic and continue our lipid discussion. So here's a giant table of fatty acids, and this is a high level science class, and I know very few, if any of you, are going into actual science labs. But one of the things that I feel that all Americans need to work on is their ability to read tables and graphs, because as a group, we are not good at understanding data presented to us. So I want to train us on how to read a graph and get, or a table, then get useful information out of it. Here's a random table of fatty acids. You tell me what trends are presented in this table. Tell me all the trends that you see. Could I start with CH3 and with COOH? Yes, that is a fatty acid. That is correct. What do you mean trend? I mean, in this data, look at all this data and tell me what happens when you change things, I guess. Like, what does this data tell you? If I just gave you all this data and I said, explain to me what's going on here, what could it be? So one trend we have is, yeah, a higher melting point with longer chain. So if you look at the first table, right, as we go from 12 to 14 to 16, our melting point goes from 44 to 53 to 63. So the more carbons, the more fatty acids. They're all acids. I mean, yeah, that's not really a trend though. What else do we have here? Yeah, even number of carbons with odd number of fatty acids. That is a trend, right? So all of our fatty acids always go up by units of 2. All of our double bonds, for the most part, let's see if I can find there are some that have even numbers in them. So it's mostly that our carbons are always our fatty acids are always units of 2. For the most part, they do start at odd numbers, but not always. Another trend, saturated versus unsaturated. Yeah. So if you look at saturated versus unsaturated, so compare 16.0 to 16.1, compare 18.0 to 18.1, compare 20 to 24, right? If you go from saturated, which we're looking up here, oh, I actually have it, to unsaturated, melting point goes down. So if you, and I'll start listing these out because more carbons, higher the melting temperature. So the more carbons, the higher the melting temperatures. What else did we say? If you're going to sat to unsat, saturated to unsaturated, melting temperature goes down. We also said fatty acids are even numbers. I have one more trend that I have written down. The unsaturated fatty acids 1N minus 9 have an. So if we have only one double bond at the 9th position, that's the melting temperature above 0. Sure, we can put that as a trend. So at least on this table, X delta 1 9 melting temp is greater than 0. Sure. Whoa, I didn't mean to draw that. I have one more. That's kind of already on there. I mean, I kind of already wrote it, but it's just a variation on that. What happens when we compare, let me get blue. When we compare 18.1 to 18.2 to 18.3, or a 24 to a 25 to a 26? So compare 18.1 to 18.2 to 18.3, 20 to 4, 24, 25, and 26. Yeah. The more double bonds, the lower the melting temperature. Oh, that's not a 26. That's a 22.6. My bad. Basically, yeah, the more double bonds you have, the lower your melting temperature is going to be. But you can see it can be complex because it depends on how many carbons are in your tail to begin with. So those are some general trends of our fatty acids. The more carbons you have, your higher your melting point. When you become unsaturated, your melting point goes down. The more double bonds you have, the melting point goes down even further. Fatty acids are even. 1 delta 9s have melting temperatures or melting points above 0. So that little exercise, I think, is very valuable in that computers can't do that, can't like verbalize that very well. So this is something humans can do very well, is look at information and pull out patterns. And that's something I hope we all can do once we leave college, is look at information and pull out patterns. Any questions about that, though? I did have a question earlier that I'll get back to right now. Somebody asked, can I re-explain the delta system? Sure. I'll do that really quick. So let's say I had this fatty acid. This would be 4,1 delta 3. The delta just tells you where the double bond starts at. So this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4. So I have 4 carbons in total. I have 1 double bond, and my double bond starts at carbon 3. That's what the delta means. All right. Moving on. This question's a little weird, so I want to explain it. Then I'll pull up a poll that will have the answer. So bacteria, and we do this too, actually, can change their fatty acid composition of their lipid membrane depending on what temperature they're at. So if you move a bacteria from cold to hot, the way they will compensate for that temperature change is that they'll change their lipids in their membrane layer. So let's say you have 2 bacteria colonies, one at 25, one at 42. What difference in their fatty acid will you likely see? So the way this answer is given, it's a weird way that these answers are given, but it's always in terms of the bacteria at the 25 degree, right? So like for A, what it's saying is that our bacteria at 25, if we compare them to 42, right? So I'm looking at the bacteria at 25, comparing them to the bacteria at 42 degrees. And what will happen, and this is answer A. I'm not saying this is the right answer, but at answer A, they'll have more 20.0 to 16.0, and they'll have more 18.2 compared to 18.0. So basically the bacteria at 25 degrees, that's what answer A is saying. People usually get confused, and this question comes straight out the book, so that's why I want to explain it a little better. But that's what it's saying for A. B, C, D, you can follow the same type of logic. I have the poll up that has this question repeated. So let's see if we can get this. I know this question is worded horribly, but those of you taking the MCAT might get questions this badly worded, so it's good to show them. I was saying A is not necessarily correct. I was explaining what A said, how to read A. Because usually when I give this question, a lot of people get confused what it's actually saying. And so I wanted to actually explain one of the answers. All right, so I'll give a couple more seconds for this. Then I'll explain the logic. Okay. So you can see here that everybody was scattered all over the place, which is a good indication that this question's not so much maybe you don't understand what's going on, but this question is horribly worded. But like I said, this comes straight out of the book. So I like to throw this in there to show you what you might face in standardized tests on information like this and how to actually read it, right? All right, let's think about these. We have a bacteria at 25 degrees C. They are cold conditions. For a lipid membrane, lipid membranes have to be fluid-like. We're going to talk more about this when we talk about membranes themselves in a few PowerPoints, but basically you want your membrane to be fluid-like. That is, if it becomes a completely like a solid, you will die. If it becomes completely like a liquid, you will die. Think of, I don't know, like a milkshake, right? A milkshake is fluid-like. It's not quite a solid and it's not quite a liquid. It's there in between. That's what you want your membrane to be. Now, if you're cold, if you're colder, 25 degrees is not that cold, but we're comparing it to bacteria at 42, right? And what you want is that you want shorter lipid tails. Because remember on the table, we said that the higher the lipid tables, the harder it is to melt. So as with more lipid carbon tails, more likely to be a solid. So as a bacteria under colder temperatures, you want shorter tails. So you want shorter tails. So you're going to have decreased levels of 20 relative to 6. So decreased 20 compared to 16. Yeah, that's one feature. Now compare 18.2 to 18, right? It's the same idea. 18.2 has a lower melting point than 18. So you don't want to form a solid at these cold temperatures. You want to be more liquid-like. So you're going to have increased 18.2 compared to 18. So the answer is D. And the shorthand answer is you want to create lipids with lower melting temperatures. So the colder you go, so the lower the temperature you go, you increase lipids with lower melting temperatures. Right? Because you don't want to form a solid. While when you increase temperatures, what you'll do, you'll increase lipids with higher melting temperatures. Because you don't want to become a liquid. It's a fine balance where, like I said, you got to find that milkshake. You know, all the bacteria, milkshakes bring something to the yard. I don't know. Convoluted answer. And it's one of those things when you look at it, you're like, what? Huh? But yeah, there's a standardized test question for you. And that's the logic they're looking for. All right, so any questions about what I just explained? The answer is D. D as in dogs. I mean, the logic makes sense because that's actually what happens. It's just, and you have to go through a couple logical loops to know what they're asking and to connect that to the information you know, which is what they ask on standardized tests all the time. Yes, and you got to answer those questions thinking fast. Would I ask questions like that on a test? I try to, you've seen two of my tests. I'm way more straightforward than this. Would, if you take the MCAT or the PCAT, will you see questions like this? Absolutely. Would the GRE have stuff on this as well? The GRE doesn't go this in depth into biology. What the GRE is, do you know how to do algebra? Do you know 1,000 random English words? At least I took the GRE in 2010. It could have changed by then. But it's how well can you write? How well do you remember random English words no one uses? How good are you at algebra, right? And so my GRE scores, I got like in the 95th percentile for knowing algebra. So I was really good at math. I got in like the 50 percentile for knowing random English words and writing essays. So I'm garbage at writing, as you probably noticed. In sciences, we don't really care how well you can write all that well. We care if you can do math. So yeah. Membranes want shorter tails and more double bonds, depending on the temperature. The hotter you get, you want less double bonds and longer tails. So the bacteria at 42, they would have more 20 zeros and they would have more 18 zeros, because the hotter you get, you want your lipids to stay together because they're fighting temperature. So you need those long tails. The colder you get, you want shorter tails and more double bonds, because what you're fighting there is freezing. So you want your lipid tails to have less connection. That's the logic. And the question I would ask if I was going to ask this on a test is, when you go to colder temperatures, how does your lipid composition change? So I would ask it straightforward like that. Well, something like this, that's what this question's asking. They're just asking it in a way that makes you really think. And that is one of the things they're testing on those tests. How good are you at thinking under pressure? All right. Let me get through this slide and then we'll call it, and we'll save the question for Wednesday as a reminder. So we were just talking about straight lipids, just one lipid. What we have a lot, so in plants and animals, not plants, so fats and oils in plants and animals, they're usually, not always, but usually what we call triacylglycerols or tags. So glycerol is our backbone. And we usually connect them to 3 fatty acids, so triacylglycerol. These are incredibly nonpolar, even though this throws students all the time. Even though we have this group, which is polar, this is just dwarfed by the tail. So the tail is extremely nonpolar, making the whole molecule nonpolar. And it will not go into water. As you have seen, if you put a fat droplet and an oil droplet in water, it won't mix. I mean, we have a whole saying in English, right? Water and oil don't mix. It's telling you right there that it's not water soluble. This is where we get our majority of energy from, which is good. Even though we don't use fats directly for energy, you can't make ATP from fat. You have to get it through glucose and the glycolysis mechanism. But since fats are water insoluble, we can carry around a lot of energy without carrying around a lot of weight. If you replaced all the fat in your body with sugar, you would weigh about 2 times more than you do right now, because sugar, it will attract water. Fats don't. So all of our energy we keep as fat, so we don't have to carry around water with it. We have different types of tags that have different purposes, and you can have different tags as in different like tail lengths. You can also have the glycerol change a little bit. When you look at fats and oils, they're really a mixture of tags. You're not going to get like an oil droplet that's all the same thing. You're always going to have variable tail lengths. When you go to the store, buy olive oil, that is just not like one type of fatty acid. You have different tail lengths as well. Plants usually have more unsaturated fatty acids than saturated. That's why it's a liquid on the store shelf, while beef fat, lard is a solid, right? And basically, fat has 6 times as much energy as an equivalent unit of glycogen. We talked about glycogen last week. Glycogen is our sugar storage. And basically, the average person, you have enough glycogen in your body to live for like 1 day. You have enough fat in your body to live for like 2 to 3 months, right? So not only is fat way more energy efficient, we can just carry it a lot more around with us easier. And your cells that have fat in them, we call them adipocytes, and they're basically just cells that just packed full of fat. Shown here. This isn't I just took off my pen. This is an adipocyte, just a fat cell. And the most annoying thing about adipocytes, this will be my last interesting fact, and I'll let you go. Once you create an adipocyte of a certain size, they really resist ever changing that size. They really don't want to shrink anymore. That's one of the annoying things. It's great for, 10,000 years ago when you couldn't really get food all that easily. So you didn't use your fat storage all that well. Nowadays, if you want to lose that fat, every time you release fat to your body to eat, 80% of it comes back to your fat cells and just gets reabsorbed. So adipocytes are a pain in the butt to shrink because we evolved that way. So once you have them, they're very hard to get rid of. That's one of the reasons why dieting is so fun. All right. Any questions about anything we went over today? If not, looks like a cold front is rolling through because my room just got about 10 degrees colder than it was when we started this talk. But I'll put up the homework. I will put this video up, of course. Yeah, that's all I have for you. The test, I believe, is sometime next week. I got asked that in Gen Chem. So let me double check for you few who are still sticking around. When is our next test? It's the wrong folder, isn't it? There it is. So our next test is November 6th, next week, Friday. All right, we have some time. Yep, next Friday, November 6th. All right. Hope you all have a good day. See everybody later.